Alright, hi SBI3U, Mr. Boudreau again. This is the second video, and actually the first video that means anything in terms of your hours for the course. I've made a few of these now, so I've improved my delivery a little bit. For example, you might notice now we've got a background around our text so you can read it a little bit better. So without too much preamble, let's just jump right into it. Here's today's lesson. Our learning goals are, first off, we're going to review the meiosis concepts. So specifically, especially the chromosome versus chromatid definition, because that's the difference between meiosis 1 and meiosis 2. Then we're going to talk about what happens when meiosis goes wrong, so abnormal meiosis, this non-disjunction stuff. We're going to talk about what happens when meiosis goes wrong in plants. Sometimes we actually want meiosis to go wrong in plants. Then we'll talk about meiosis going wrong in humans and some of the diseases that it can lead to. We'll talk about amniocentesis and karyotyping, which is how we detect those chromosome abnormalities before the baby is even born. And then finally, assisted reproduction technologies. So certain things that can help parents that want to have a baby that might have some kind of biological issues that prevent them from having a baby. Alright, so I'll pull up the current topic. That's a review of meiosis concepts. And we are going to jump over to, let's pull that away. We are going to jump over to the desktop. And we're going to be looking at this website here. This is called Cells Alive Interactive Meiosis. You can find a link to that down below in the description underneath all of the timestamps. And yeah, keep in mind, timestamps are great. If you're watching this video again and you want to jump to a specific section, just check under the video and you'll be able to click on the timestamp. Okay, so let's review this meiosis thing. I'm going to play it at full speed. Just so you see it all in one shot, and then we're going to go step by step. Notice that it's going to flow through the phases here as the virtual cell hits those phases. So yeah, let's just watch it full speed first. Okay, I'm going to turn off the sound because that was really annoying. Yeah, so full speed, you can see that there's a lot going on here, but the whole point is we start with one cell, and we end up with 4 cells. The one cell is diploid, a diploid cell, meaning that it has 2 chromosomes, and the gamete, each gamete is autoploid, we call it autoploid, which means it only has 1 set of each chromosome. But it has to go through all of these stages first. So let's get into the stages. Alright, let's get into these stages. First off, we have this cell. Because it's not at any of these phases, we assume it's in interphase, which means it's just doing normal everyday cell stuff. It's replicated its DNA so that it's ready to go. So it's actually got twice the DNA that it normally has before it goes through this whole process. The first thing that will happen, well, 2 things happen simultaneously. First, the chromosomes are going to go from this kind of spaghetti stuff into, you can see them kind of packing up from being spaghetti. So, spaghetti, organized. And the second one is you're going to have these microtubules start forming because eventually those are going to pull the thing apart. So both of those happen at the same time until you get into prophase, and now they're packed up. Near the end of prophase, you'll see the nuclear envelope dissolve. So before it was a nice envelope, after it dissolved. And right here, this is an interesting place to stop because you can see the crossing over event happening during prophase 1, kind of pro-metaphase 1. That's when you can sometimes get a swap. You can see them swapping there. So crossing over, and then they swap, and now they're stuck like that. Right, they've done crossing over, they're slowly lining up, they line up in the middle, and they actually get turned to be perfectly lined up in the middle, and now we're fully in metaphase 1, because now the microtubules are going to pull them apart. I'm going to pull them apart, and we'll see that happening slowly. Off they go through anaphase 1. Anaphase 1 is very quick. Off they go. And then a lot of stuff happens all at the same time. The microtubules dissolve. So from here to here they dissolve. The nuclear membrane starts to reform, and the sides of the cell are going to pinch in through cytokinesis, and it's going to turn into 2 cells. Now interestingly, this centromere up here is going to now split into 2 kind of sub-centromeres and come around to the sides. You see that? So there was one before. Well, there were 2 of them before because they pulled them to opposite sides, these 2. And then they split to make new microtubules. And now we're in prophase 2, and the same thing starts to happen. You see that they used to be in a nuclear membrane, and then they get unpacked, and that nuclear membrane dissolves. Then they line up, like you expect, to metaphase 2. And then this is the difference between meiosis 1 and meiosis 2. Back in meiosis 1, right, we had, you can see that there's 2 sets of each chromatid. These are individual chromosomes, but they have homologous chromatids, right? They're still in that kind of X shape that you see often in chromosome pictures. But when we get down to metaphase 2 and it's time to split them apart, they're no longer paired up. They're just individual chromatids. There's no homologous chromatids going across. So that's the difference between meiosis 1 and meiosis 2. If you remember from our learning goal, that was part of it, chromosome versus chromatid. In meiosis 1, the chromosomes separate, but the chromatids stay together. In meiosis 2, those chromatids also separate. So please be aware of the difference between those 2 terms. Wink, wink, nudge, nudge. You know, it's probably going to be on a test somewhere. Okay, continuing on. The same thing happens, telophase 2, very quickly the microtubules dissolve, the nuclear membrane reforms, and then cytokinesis pinches it off, and now we're down to individual gametes. We have cut the amount of DNA in each gamete by a factor of 4, but the number of chromosomes has only been cut by a factor of 2. That's why it went from 2n to 1n. Yeah, so that's making sperm or eggs. The only kind of little fiddly bit is sperm, spermatogenesis. All 4 of these will go on to become sperm cells, but in oogenesis, when you're making eggs, only one of them, I'm not sure how the body decides which one, but one of them becomes an egg, and the other ones just get dissolved and recycled for their parts. You know, I guess there's only enough resources to make one like really nice egg. Meanwhile, sperm are just getting produced like crazy because you need millions of them. Okay, so that's a quick review of meiosis, and the point of meiosis, of course, is making gametes. So let's jump back to the front camera and review this. Yeah, we have done our meiosis review. That's done. Let's move down to abnormal meiosis, which means I'm going to have to jump over to the PowerPoint. So back to this view, and we're going to go into the PowerPoint. So here's the PowerPoint that we're working on today. It is called abnormal meiosis or fertilization. Yeah, so obviously very related to what we're talking about. It's number 4.4. You'll find it in your EDSB. Let's jump in. So first things first, meiosis is delicate. You saw just how much has to go right. Those tubes have to attach at the right place. They have to grab the right thing and pull it apart. And so there can be errors, lots of different errors. Sometimes it's in the crossing over, sometimes the crossing over doesn't go well, and you actually end up with part of the old chromosome on the new chromosome, so you get like parts that are attached where they shouldn't be. Sometimes it just doesn't work at all. Sometimes the gametes don't split correctly, and then they just get tossed. There's all kinds of interesting cell mechanisms that will stop the process if it's not going well. So yeah, there can be errors, and if it happens during fertilization, you know, if that specific gamete that has the problem ends up actually being the thing that forms a zygote, you know, maybe mom has a problem making one particular egg, and that one particular egg out of thousands of eggs is the one that ends up actually getting fertilized by a sperm, then you're going to have genetic problems in the baby. Or same thing with the sperm. If dad has a problem making sperm correctly, and it's that one sperm that ends up fertilizing the egg, the zygote is going to have genetic problems. And because it happens so early, because it happens in the fertilization process when it's just one sperm, one egg, and then that's going to grow into a whole baby, that means that the entire human that gets formed is going to have that genetic problem. It's not the same thing as like a tumor where it's just in one place. It's going to be the entire baby. Okay, so let's go to the next slide here. So nondisjunction is probably the most common way that these genetic disorders come about, and it occurs somewhere between metaphase and anaphase. And if I jump back to our interactive meiosis thing, right, that's between here and here, somewhere between these two, right, metaphase and anaphase. You know, metaphase and anaphase, somewhere between them lining up and them getting pulled apart, something just goes wrong. Yeah, metaphase and anaphase, metaphase and anaphase. So something just goes wrong in that thing. And what happens then is it's called nondisjunction, meaning that both of them go to one place. You know, both of them end up in the same place. Instead of doing this, they're doing this, which means that one daughter cell has an extra, and the other one over here has none. So you actually end up with two bad eggs or two bad sperm out of the deal. Okay, so a question for you guys. Do you think this happens in meiosis 1, in meiosis 2, or in both? So going back to a normal meiosis, this is normal meiosis. All right, there's this metaphase to anaphase transition. Then there's this metaphase to anaphase transition. And the question is, which one of these transitions will create a gamete that has too many or too few chromosomes? So that's the question that I'm asking right now. I'm going to count down from 5. Pause the video if you want to take a minute to think about it. And I'm going to count down. 5, 4, 3, 2, 1. Okay, so the answer is both. It can happen in both. Here's a diagram of it happening in both. On this side, you see this blue arrow. This blue arrow is showing the nondisjunction, and it happened during meiosis 1. Sorry, this one happened during meiosis 2. This one happened during meiosis 1. So there's these red chromosomes in the egg. So they both start with the same, you know, 2 red chromosomes in the egg. Meiosis 1 will split those 2 red chromosomes into their homologous pairs, normally. But on this side, both of them went one way. Right? Both of them went one way. But on this one it went normal, except for in meiosis 2, this one split unevenly, so there's a nondisjunction here. This one went okay. So this is what it should normally look like after it gets fertilized by sperm that are carrying the blue homologue of that chromosome. So that's what it should look like normally. Assuming all of these sperm are normal, these become normal cells. This one becomes gets a trisomy. This one gets a monosomy. This one gets a trisomy. This one gets a trisomy, and both of these get monosomies. So if it happens earlier in meiosis 1, you get 4 bad eggs. If it happens later in meiosis 2, you just get 2 bad eggs, but it can happen in either one. And you wouldn't be able to tell looking at these eggs. Like if you did a genetic test on a baby, you wouldn't know whether it happened during meiosis 1 or meiosis 2, because all you can see is that one egg that turned into that zygote. So yeah, ooh, I haven't changed my my review, so Nondisjunction. Okay, so that's nondisjunction. Right, and there's some key terms here. Right, it'll result in either 22 or 24 chromosomes. Remember that normally we have 23 homologous chromosomes, and our normal chromosome number is 46. So it should be 47 or 45 chromatids is what that should really say. Yeah, 24, that's trisomy, 3 of a chromosome. 22, it's monosomy, it's a single copy. So trisomy, monosomy, I think that's pretty self-explanatory as a term. Okay, chromosome number. All right, chromosome number, like I said, 47 or 45. 46 is the normal chromosome number in a human cell. Sometimes we actually want this. So this is plant. Right, plant polyploidy, which is, of course, one of our learning goals. Right, we've done review meiosis concepts. We've done abnormal meiosis, this idea of non-disjunction. Important there was that it can happen in meiosis 1 or meiosis 2. If it happens in meiosis 1, all 4 gametes are affected. If it happens in meiosis 2, only 2 of the 4 gametes are affected. But in both cases, you have an equal number of, too many and too few chromosomes because of the non-disjunction. Okay, next up, we're in ploidy levels in plants. So sometimes this is actually useful for us. Sometimes we have we actually take advantage of plant polyploidy. So, for example, a lot of our cultivar plants, a lot of our plant cultivars are polyploid. Diploid is normal, but your typical seedless watermelon or banana, which is also seedless, are triploid. Blackberries and peanuts are tetraploid, so they've actually ended up with 4 copies of the chromosome where they should have 2. Sweet potato and wheat are normally hexaploid. Some wheat strains are actually dodecaploid, so they've got 12 instead of 6. But yeah, normally it's 2. And then strawberries, notably, are octaploid. They have 8 copies of every single chromosome. For some reason, in plants, when they have multiple copies of a chromosome, they get bigger and sweeter fruit. So we like having that as a thing. And triploid, we can actually force a watermelon to be triploid, and here's how they make seedless watermelons. If you've ever been curious, like, the things have no seeds, how the heck can we actually grow one? Well, what they do is they take a tetraploid watermelon and a normal diploid watermelon, and they pollinate one with the other, and then the tetraploid, because it's got 4 copies, it's going to send 2 to its pollen. The diploid, because it's got 2 copies, is only going to send 1 to its egg cell, or whichever combination they want. And so 2 from mom, 1 from dad, you get 3, so all of your offspring are triploid. And then for some reason that just makes them not make seeds. They can still get fertilized, but they don't make seeds in their own fruit. So we can kind of force triploidy for the purposes of making seedless watermelons, which I think is kind of cool. We can also force other types of polyploidy with certain chemicals. So, for example, there's this chemical called calicine, I think, calicine, there we go. This chemical, it stops microtubule development, and if we go back to here, right, the thing that actually pulls these things apart are these yellow microtubules. So if you stop those from forming, the cell will still go through this process, except it'll end up completely tetraploid or completely diploid or whatever. So a lot of plant research is actually focused on finding these useful polyploid plants, and you can actually have you can treat one side of a plant with calicine and colchicine. Treat one side of a plant with colchicine and the other side without it, and you can actually end up with a plant that's diploid on one side and tetraploid on the other, which is really, really neat. And you can take that and plant it, and then your whole plant is going to end up tetraploid if it's making a good fruit, something that you want to see. You know, bigger leaves, bigger fruit. For some reason, plants just do that when they're multiploid. Cholecystine. I'm going to say cholecystine. Cholecystine is also used as a human medicine, so it's used for like gout and stuff like that. A lot of inflammatory stuff, because microtubules are involved in inflammatory stuff, and you know, you definitely get warnings not to take it if you're pregnant, because it messes with microtubule development, and so it'll mess with your meiosis. So that's plant polyploidy. Jumping back to the front cam and pulling up this, we've done polyploidy in plants, ploidy levels in plants. Next up, we're going to talk about nondisjunction in humans. So why does it occur, and what kind of effects can we have in nondisjunction in humans? So let's change our current topic to nondisjunction in humans. Right, so we'll save that, and we'll do a keep in mind here. The keep in mind for nondisjunction in humans is not all of the eggs and sperm are affected, only some. So not all the eggs and sperm are affected, only some of them get affected by this. So moving on to nondisjunction in humans. Okay, back we go to this view. You don't know this view, I like that one better. And we're jumping into nondisjunction in humans. Nondisjunction in humans, you can see this is called a karyotype. So karyotype is a keyword. It's all of the chromosomes spread out on a piece of plastic. You can actually pay for services that do this for you. You get a little cheek swab and you send it in, and they'll lay it out for you and take a picture of it if you want a cool nerdy picture of something. So that's a nondisjunction. That's a karyotype. And you can do this before birth. You can actually go and get some of the baby's DNA before the baby is even born. That's prenatal testing. This is recommended for women over 35 years old because of the loss of this protein called cohesin, which is this really cool set of proteins. You can see the picture of it down here. These gray things are the two homologous DNAs. They're doing the spiral thing that DNA does, and they're being held together by like a lasso of protein. Those are the lasso, and that's what actually holds the two homologous chromosomes together, the chromatids together in meiosis. That makes them move together as a unit. And it's also where the microtubules are going to attach. All right, there's this kind of an attachment point here and here, and that's where they're going to grab them and pull. So it's like a little attachment point and pull. So what happens to women that are over 35 years old is as the eggs have been sitting there, because women don't make more and more eggs, they just use the same batch of eggs and draw from their egg supply. So those eggs that they're using after they're 35 have been sitting there in their ovaries for longer, and slowly that cohesin goes away. And then it's holding this less tightly, so it's more likely for there to be weird nondisjunction stuff happening. That maybe the microtubule won't be able to attach onto here, or maybe both of them will go. Maybe this won't detach. It's just issues with that cool little protein lasso that's holding the chromatids. Right, that's nondisjunction in humans. Next up, we're on to our next topic, which is amniocentesis and karyotyping. So let's jump back into it. I'm going to change this, amniocentesis. I'm probably spelling that wrong. Amniocentesis and karyotyping. Amniocentesis and karyotyping. Okay. Let's pull that away. We'll jump back into our our nice this view. I like that view. And let's talk about amniocentesis. Oh, I did get it right. Amniocentesis and karyotyping. So here's how this works. This is how the prenatal genetic testing works, and it's a fairly simple procedure, and it's considered non-invasive because it's just a small needle that goes in. So here you see an actual picture of it being done, and here's a diagram of what it looks like. Right? The baby has to be big enough for there to be like a big enough amniotic sac. The amnio part, amniotic sac, is where the amniocentesis name comes from. So there's a little cognate for you if I ask you what amniocentesis is as a technique on a test or something like that. And what they do is they just stab into there with a needle. You know, it's got to be kind of freaky if you're the mom, but it's a useful test, so I assume that that they're okay with it. To avoid stabbing the baby, they have an ultrasound going at the same time. So just like you get an ultrasound to see your baby, they use an ultrasound and they can see the needle going in. And they just draw out some of these some of the amniotic fluid, and in that fluid is going to be floating some of the baby's cells, because you know fetuses are actually alive in there. They're moving around, they're drinking, they're they're actually listening, they're peeing and they're pooping and stuff. So there's cells in that fluid that they can use to do a genetic test. So that's how they do a karyotype. That's how they get a karyotype. Right? So they might do that. They might pull something out, and they'll have this karyotype. Right? And the question is, what's wrong? So I'll do the same 5-second thing. Pause the video if you need more than 5 seconds. But here we have the chromosomes, always the sex chromosomes at the end. So this baby is a little girl, which is nice. Yeah, I'm going to do the 5-second countdown. 5, 4, 3, 2, 1. And what's wrong here is here. There are 3 chromosome 21s. So that's called trisomy. I'm going to change my keep in mind. And that's 3 chromosomes equals trisomy. 1 chromosome equals autoploidy. Well, monosomy, sorry. Right. 3 chromosomes is trisomy, 1 chromosome is monosomy. Okay, so yeah, what's wrong here is there's 3 of chromosome 21, and that's called Down syndrome, named after some researcher named Down, who was the first one to describe the symptoms very well, I guess. A lot of these syndromes are just named after the people that discovered them, like Alzheimer's is named after Alzheimer and stuff like that. Okay, what's wrong here? Same thing, I'll do the countdown. So don't just tell me what's wrong, also tell me what gender or what sex, what biological sex this fetus is going to be. So 5, 4, 3, 2, 1. Okay, so first off, the gender or the sex. This is a boy because there's 1 X and 1 Y, so clearly that's going to be a boy. And this boy has 3 copies of chromosome 18. So what do we call that? We call that trisomy 18. Or if you want to use, an old white guy's name, it's Edwards syndrome. Again, Edwards, the guy's last name was Edwards, so it's Edwards. No apostrophe, just Edwards syndrome. All right, here's a few other ones. Trisomy 13 is called Patau syndrome. But there are some that just involve the sex chromosomes. So for example, Turner syndrome and Klinefelter syndrome. Turner is interesting because there's only 1 X and there's no Y, which means that Turner is a monosomy. If you think up here, a monosomy in X. And it's, I guess, the only monosomy that's supported in humans. It's the only monosomy where you can actually have a viable baby. Klinefelter is 2 X's and a Y, so that's a trisomy, but it's XXY. Yeah, you can also have other ones like XYY, so that's like, it's called a super male, which is kind of cool. Yeah, so those are some different karyotype disorders that you can get as humans. Let's briefly talk about the rate of these problems. Yeah. So we have to. I'm gonna move over to the other side. Let's talk about the rate of these problems and the kind of some stats about issues in fertilization and in childbirth and stuff. So first off, about 10% of couples have fertility problems, and this isn't just like, they try it once and they can't conceive. This is like consistent effort multiple times and they can't conceive. So that's what fertility problems are defined as. It's split about 1/3 male, 1/3 female, so it's not like it's one person's fault. And roughly 1/3 are because of both or some other problem. Some of the issues here, you could have reduced sperm and egg count or quality. Sometimes, those gametogenic cells are just not great. Sometimes the tubes, the fallopian tubes that the egg travels through, sometimes they're blocked. And so the egg never makes it down to the uterus and can't get fertilized because it's not in there. Or the same thing in the males, the blocked epididymis or the blocked vas deferens. Some people do that on purpose, right? You get a vasectomy. What they'll do is the vas part of vasectomy is just referring to the vas deferens. They will block that tube on purpose. Now they're actually working on kind of a temporary solution where they kind of inject a thing that blocks that tube, and then they can take that away instead of having to have surgery instead. It's kind of interesting. We do this to our pets too, our cats and our dogs. Like my cat has had the neutering operation, and my dog has had the spaying operation, and it's the same thing. It's just either blocking the tubes or just completely removing the gonads, as they did for my cat. Yeah, so that's some possible causes there. Another possible cause, which is about 15% of all miscarriages, is called polyspermy, and that's what happens when two sperm end up fertilizing one egg at the same time. That's automatically basically a miscarriage. I believe there was, I was reading about this, there was one case of a baby that had that and survived until 7 months, but never developed. Just stayed like a big baby for 7 months and then died from some kind of immune problem. So yeah, polyspermy is like all the trisomies together, because you'll have 3 copies of every single chromosome. It's just bad. Back to some other stats. 30 to 50% of all fertilization events end in miscarriage. So, up to half of all fertilizations. And that's all of them, right? Every single fertilization. So you can get fertilized without knowing that you're pregnant, because you just get a miscarriage. And if it happens early enough in the process, it just kind of comes out, and a lot of people just completely miss it. So yeah, it's a big number. It's a very delicate process, and there's enough checks and balances to make sure that it doesn't go wrong, that literally up to 50% of all possible fertilizations just don't work. But known pregnancies, once it's actually gotten to the stage where you can test positive on a pregnancy test, about 10 to 20% will end in miscarriage, and this goes up considerably with age. So young people, it's about 10%. Women over 40, it's like half of their pregnancies are likely to be miscarriages. So yeah, and it goes up with age for the same reasons I talked about earlier, which is like that protein cohesin, right? As the eggs sit there longer, they will lose some of these vital proteins that make the fertilization go well, that make the meiosis go well. Alright, so that's that. Let's go back to the front. let's pull up our learning goals again. We've done amniocentesis and karyotyping, one of the most common prenatal tests that helps parents figure out, find out if their baby has any kind of chromosomal issues. That test was actually popularized by a Japanese Canadian. She did a lot of work with it. It's a interesting story. If you want to look that up, I'll have a link to her biography down below the video. Okay, next up then, and the last thing, is assisted reproduction technologies. So we're going to talk about some of the different technologies that help women and men conceive if they're part of that, part of the fertility problems type group, and talk about some of the regulations about these technologies that we have here in Canada. So let's change our current topic then to assisted reproduction. Assistive reproduction tech. Alright, assistive reproduction tech. And keep in mind this technology, keep in mind 10% of Ontarians experience reproductive fertilization problems. I should really start just pausing the video when I do these. Right, so 10% of Ontarians experience fertilization problems. So these technologies are for those 10% of people. And also keep in mind that the older you are, the higher the chance gets. So it's usually used to help older people as well. So let's jump into some assistive reproductive technologies, or assisted reproductive technologies, ARTs. There's a few different ones, and they enhance the chances of successful reproduction. So specifically, they tend to try to improve whatever aspect is lacking in the couple's fertilization goals. So for example, if there's a low sperm or a low egg count, there are drugs that will increase sperm and egg, sperm production, and they can increase the release of the eggs. So multiple eggs get released at once instead of just one, and then you have a better chance of one of them getting fertilized. You could also select the best gametes. Not every sperm and not every egg is the same because of the little differences that can happen in meiosis. So you could actually pull out the sperm and the egg, check it genetically, and then put two together that should work, and then put them back in. You can also increase the chances of fertilization, so you can get drugs that, like I said before, they increase the release of the eggs, or they'll increase the speed at which it moves through the tubes or something like that. And you can have drugs that increase the chance that the zygote itself implants onto the uterine wall and forms the placenta and all that stuff. So you can there's drugs to support and technologies to support every step of the process, essentially. So they're usually used to make up for whatever is lacking. So you might have heard of this stuff, IVF, in vitro fertilization. What happens there is the woman gets a drug that increases how fast she makes eggs. And then there's a surgery where they actually go into the ovaries and they pull out those eggs. Those eggs are fertilized externally with some sperm from someone. They just choose somebody. If it's a husband and wife, maybe the husband donates the sperm. If it's just a mom that wants a kid, they might get it from a sperm donor. But they'll fertilize it externally, and then you might have heard of test tube babies. Well, this is literally a test tube baby. They will fertilize it, grow it for a little bit to make sure that it's developing okay, and then they'll put it back in the mom, in the uterus, and they'll implant like a normal zygote and have a placenta and a belly button and all that stuff. This isn't like cloning or anything. It's just taking the normal reproductive stuff, the sperm meeting the egg, and just doing it outside. So this is interesting. They'll often implant 2 or 3 zygotes because not all of them take, and so you'll often get people with IVF that have 2 or 3 zygotes implanted, and then they end up having twins or triplets, which leads me to an interesting news story, which is here. You might have heard of Octomom. I don't know if you have, but this is a news story from 2011. And what this was there was a woman who had some of her eggs, some embryos that were ready for implanting, and she asked her doctor to implant a lot of them, and he ended up implanting 12 embryos, which is kind of irresponsible. As said here, the medical board said he did an excessive number of embryo transfers into Nadia Suleiman, and she ended up having 8 of them implant, and so she gave birth to 8 kids. Yeah, it's kind of crazy, right? So extreme departure from standard care, and the doctor he lost his license. He got fired because of this. Yeah. And at the start, this was a big story, the Octomom story, because she's having 8 kids. I mean, it's a big news story. But then people found out she already had 6, and that she was a single mom and that she was unemployed and she was on welfare and stuff. And then the public opinion kind of turned against her. But she's doing well now. She's like a blogger or something. She does some kind of lifestyle thing with it. It's kind of cool. If you're interested, they have a Wikipedia page, and it even says when they were born. Right, they were basically born like a minute apart, and they had a very low birth weight, which is of course one of the concerns when you have such a big multiple pregnancy. Yeah. Okay. So that's that. Let's jump back to this. So that's IVF. It has to be carefully done. You need to watch out for how many you put in there. So that's IVF is pretty common. Artificial insemination is also common, especially when you have, for example, same-sex couples that want a baby. You can just do that. You take sperm cells and just artificially inject it into the uterus. So presumably that's done with the best quality gametes. You select the sperm that don't carry the trait that you're looking at, and then you only let those into the uterus so that the baby has no chance of getting that trait. Okay, so we talked about the doctor losing his license. We actually do have an act here in Canada, a piece of legislation that restricts some of these things. So in particular IVF. This is called the Assisted Human Reproductive Act. It came out in 2004. It's actually a really good piece of legislature. Canada has a very comprehensive kind of law about this. So there's some prohibitions here. For example, we can't use IVF embryos for anything other than implantation or training. So you can't take those embryos and steal their stem cells and stuff like that. You definitely can't clone humans, so that's prohibited by law. I mean, you can very easily clone humans, where we have the capacity. It's just legally no. You also can't sex select human embryos. So if you're a mom and you're gonna get some embryos implanted, you can't say put 3 girls in there. It has to be random. But there's an asterisk there. If you're carrying a Y-linked or an X-linked trait and you don't want that trait, specifically Y-linked traits, you can ask for only girls if there's a Y-linked trait that you're trying to avoid. So yeah, that's the human embryo thing. You can't implant or harvest without consent. I mean, that just kind of goes without saying. You can't do any weird cross-species stuff, so you can't try to implant a cow embryo in a human or a human embryo in a cow or any kind of thing like that. So there's like a whole chunk of the act that's like, yeah, can't do any of this. Also, here's an interesting one: is you can't pay anyone for sperm or eggs or embryos or surrogate mothers. This is a Canadian thing. In the States, you can actually get paid to donate sperm and get paid to donate eggs and stuff like that, and they can pay their surrogate mothers, but in Canada, that's not allowed. You are allowed to pay for like the hospital stay for the surrogate mother. You're allowed to pay for anything related to the pregnancy, but you can't just pay them just to be a surrogate mother. You can't even like cover their pays, like the pay that they miss being pregnant, unless that was ordered by the doctor. Unless the doctor told them that they had to stay home for the health of the baby, then you can cover their paycheck and only their paycheck. So yeah, there's all these rules about not paying surrogate mothers. And there's also some things that it controls. So for example, surrogate mothers have to be at least 21. There's a minimum age. I guess they figure that's the age that you can make that decision to be a surrogate mother. And sperm and egg donors have to be over 18. So you can't take sperm and egg from anyone that's under 18. I guess that's part of the consent piece. There is an asterisk there too, and that's if the person that's under 18 is going to be going through some kind of treatment like chemotherapy or something like that, and they want to save their sperm and egg for their own personal use later. So if you're under 18 and you're producing sperm and eggs and you have some kind of medical condition that might impact that, you can actually save those and then use them to get pregnant later, I guess. So those are our kind of laws about this stuff. All right, so back to our learning goals. We have reviewed meiosis concepts using the website, the Interactive Meiosis website, which is that website there. That'll be in the description. We've reviewed some abnormal meiosis of some nondisjunction stuff. What causes nondisjunction, those microtubule things. We've talked about plant polyploidy, like how you make a seedless watermelon. We've talked about nondisjunction in humans and some of the risk factors for that, in particular age. We talked about amniocentesis, the method of pulling out the baby's DNA straight from the womb without affecting the baby. We talked about karyotyping, laying out those chromosomes to make sure that they're okay. And we've talked about assisted reproduction techniques, assisted reproductive technologies, and the Assisted Reproductive Act of 2014. So that's the end of this lesson. Keep an eye out for its quiz on SBI. And this one does actually count, so please do that quiz, and I'll see you guys in the live class on Monday at 8:40 for any questions about this stuff. And there's also an assignment here to do about karyotyping. I'll talk about that during the live session. Thank you very much.